Hardy-Weinberg Equilibrium, HWE. Part 1. UGC-NET Anthropology. Welcome to AnthroNet Academy, a study aligned with the UGC-NET Anthropology syllabus. Description box. Please visit the description box for books references. Dear learners, if you find this content helpful, please consider subscribing to AnthroNet Academy. Your support really means a lot and keeps me motivated to create more valuable content for you. Big surprise at 500 subs. Once AnthroNet Academy hits 500 subscribers, we're dropping something truly special just for you. A full video revealing 100 powerful, underrated, and must-read books list in anthropology, complete with author names and insights important for UGC-NET exam. Test yourself. Don't miss the final segment of this video. It includes high-yield MCQs that could make or break your next anthropology exam. Mendelian population. It is defined as an interbreeding group of organisms which share a common gene pool or a reproductive community of sexual and cross-fertility individuals which share a common gene pool. Gene pool. A total of genes shared by reproductive members of a population, or we can also say the collection of all the alleles in a population. Phenotype. External appearance of an organism resulting from the interaction of its genotype and its environment. Genotype. The genetic makeup of an individual at a given locus defined by what has been inherited from both parents. Allele. An alternative form of a gene or DNA. Locus. A specific location of a gene or other DNA sequence on a chromosome. Introduction. Most genetics research focuses on the structure of genes on chromosomes, the function of genes, and the process of genetic transmission from parent to offspring. Population genetics instead focuses on the overall gene pool in a population of interbreeding organisms, that is, the frequency of all alleles of all genes in the population, and whether the gene pool may be changing across generations in a population. Population genetics examines the relationship among genotype frequencies in a population, allele frequencies in its gene pool, and factors that can change these frequencies over time. History. The Hardy-Weinberg principle was discovered independently by G.H. Hardy, a British mathematician, and W. Weinberg, a German physician, in 1908. According to this law, allele and genotype frequency in a population do not change from one generation to another and remain constant. This principle postulates a set of conditions in a population where no evolution occurs. Genetic equilibrium and the Hardy-Weinberg principle. A population is in genetic equilibrium when allele frequencies in the gene pool remain constant across generations. A gene pool will be in equilibrium under the following conditions. The population is very large. Individuals in the population mate randomly. There is no migration into or out of the population. Natural selection does not act on any specific genotypes. Males and females have the same allele frequencies versus individuals are diploid and reproduce sexually. No mutations occur. Mathematical representation. This mathematical model is a powerful tool for developing evolutionary models, and the law is P2 plus 2 PQ plus Q2 equals 1. If we have a locus with two alleles, A and A, where P is the frequency of the A allele and Q is the frequency of the allele. Both Hardy and Weinberg showed that the expected genotype frequencies in the next generation are Expected frequency of genotype AA equals P2. Expected frequency of genotype AA equals 2PQ. Expected frequency of genotype AA equals Q2. If we have A equals P equals 0.6 and A equals Q equals 0.4. From these allele frequencies, we can figure out the expected distribution of genotypes using the Hardy-Weinberg Equilibrium in the next generation as expected frequency of genotype AA equals P2 equals 0.6, 2 equals 0.36. Expected frequency of genotype AA equals 2PQ equals 2, 0.6, 0.4 equals 0.48. Expected frequency of genotype AA equals Q2 equals 0.4, 2 equals 0.16. Note that the sum of these genotype frequencies adds up to 1, which they do here. 0.36 plus 0.48 plus 0.16 is equal to 1.0. Applications of Hardy-Weinberg Equilibrium. 1. Detecting evolutionary forces. If observed genotype frequencies deviate from expected values, it suggests that evolutionary forces, like selection or migration, are acting on the population. 2. Estimating carrier frequencies. Useful in medical genetics to estimate the proportion of heterozygous carriers of recessive genetic disorders. Example, in sickle cell anemia, if 1 in 10,000 individuals is affected, homozygous recessive, you can estimate carrier frequency using 2PQ, 2PQ, 2PQ. 3. Forensic and population studies. Helps in DNA fingerprinting, establishing population structure, and understanding gene flow among human populations. 4. Anthropological significance. Used to study the genetic structure of tribal, caste, or isolated populations. Can reveal inbreeding, genetic drift, or migration patterns. Mating patterns. To maintain a population in Hardy-Weinberg equilibrium, 5 key assumptions must be met. Violation of any of these assumptions can cause allele or genotype frequencies to change over time, meaning evolution is occurring. 1. Large population size, no genetic drift. Explanation. The population must be infinitely large to minimize the effects of random changes in allele frequencies, known as genetic drift. Violation effect. In small populations, chance events, like random deaths or births, can significantly change allele frequencies between generations, leading to loss of genetic variation. 2. Random mating. Explanation. Individuals must mate randomly, without preference for certain genotypes or phenotypes. Violation effect. Non-random mating, e.g., inbreeding, assortative mating, can alter genotype frequencies, especially increasing homozygosity, but allele frequencies may remain unchanged. Leads to Hardy-Weinberg disequilibrium without necessarily causing evolution. 3. No mutation. Explanation. There must be no new mutations altering existing alleles or creating new ones. Violation effect. Even a small mutation rate can slowly introduce new alleles or alter allele frequencies, especially over long timescales. Mutation is the ultimate source of genetic variation. 4. No migration, no gene flow. Explanation. There must be no movement of individuals and their genes into or out of the population. Violation effect. Immigration or emigration can introduce or remove alleles, changing the gene pool. leads to genetic mixing, especially significant in human populations. 5. No natural selection. Explanation: all genotypes must have equal chances of survival and reproduction. Violation effect. If certain alleles increase fitness, they will become more common, violating equilibrium. Selection alters allele and genotype frequencies over time. This is the main driver of adaptive evolution. Summary table. Assumption explanation if violated. Large population prevents random changes. Genetic drift. Drift affects allele frequencies. Random mating ensures equal mating probabilities across genotypes, alters genotype, but not allele frequencies. No mutation, no new alleles, introduce changes allele pool over generations. No migration gene flow is blocked, new alleles may enter or leave population. No selection, all genotypes are equally fit, fitter alleles become more frequent. Test your knowledge. Q1. In a population, if the allele frequencies remain constant, but genotype frequencies deviate from Hardy-Weinberg expectations, which assumption is most likely violated? A. No mutation. B. No migration. C. Random mating. D. Large population size. Correct answer: C. Random mating. Explanation: deviation in genotype frequencies while allele frequencies remain stable usually indicates non-random mating, e.g., inbreeding. This increases homozygosity but does not necessarily change allele frequencies. Q2. In a population where 4% of individuals express a recessive trait, what percentage of the population is heterozygous, carriers, assuming Hardy-Weinberg equilibrium? A. 16%. B. 32%. C. 36%. D. 48%. Correct answer: B. 32%. Q3. Which of the following statements is false regarding Hardy-Weinberg equilibrium? A. It assumes infinite population size. B. It allows for mutation at a constant rate. C. It assumes no differential reproductive success. D. It can be disrupted by gene flow. Correct answer: B. It allows for mutation at a constant rate. Explanation: HWE does not allow mutation at any rate. Even a constant mutation rate violates the assumption. All other options are true under the HWE model. Q4. If the genotype frequencies in a population are 0.36 AA, 0.48 AA, and 0.16 AA, what is the frequency of allele A? A. 0.36. B. 0.48. C. 0.6. D. 0.76. Correct answer: C. 0.6. Q5. A small, isolated human population shows rapid changes in allele frequencies over generations despite no evidence of selection, migration, or mutation. What is the most likely cause? A. Non-random mating. B. Founder effect. C. Heterozygote advantage. D. Balanced polymorphism. Correct answer: B. Founder effect. Explanation: the founder effect is a form of genetic drift common in small, isolated populations. It can lead to random changes in allele frequencies unrelated to selection. Thank you for watching. Please like, share, and subscribe to my YouTube channel, AnthroNet Academy, for more insightful content on anthropology. Stay safe and connected.